Hi everyone, and welcome to our video on gene mutations. This is the first video in our Module 6 Part 1 series looking at cellular control. And in this video, we'll be looking at specification reference 6.1.1a, types of gene mutations and their possible effects on protein production and function. To start off with then, let's remind ourselves of something that hopefully we all learnt back at GCSE, the definition of a mutation. So when we're talking about a mutation, this is just a random change that occurs to the genetic material. Now, by that, what we could mean is that we're seeing some kind of a change to either the chromosome structure or the chromosome number of an organism, or if we're talking about a gene mutation, this is where we're seeing an actual change to the DNA itself. In terms of what's going to cause these mutations, first thing to bear in mind is that these just happen spontaneously during that process of DNA replication. It's not perfect. Errors happen. Now, obviously a number of those will be corrected, as we saw with that whole process back in Module 2, looking at the cell cycle and those checkpoints, but some get through. So we do have that spontaneous occurrence of these mutations during DNA replication, but we will have certain things that increase the likelihood of a mutation occurring. And these are things we refer to as mutagens or mutagenic substances. And a couple of good examples that you've already encountered in probably your physics back at GCSE, ionizing radiation, and if you're a smoker, tar that's going to sit in your lungs. Again, not good. Don't smoke. Let's move on then to think about what kind of mutations we could see. Now, if we're getting a mutation taking place during the process of mitosis, this is what we refer to as a somatic mutation. And these somatic mutations are the ones that are happening in our regular body cells, because hopefully we remember that when we're talking about mitosis, this is how we're going to be replicating our regular body cells. So somatic is just referring to our normal body cells. Because it's only affecting certain body cells, this will not be passed on to any offspring, because obviously the only way that we can affect the genomes of the offspring is by having some kind of alteration to the gametes. This is a normal body cell, therefore no alteration to the gametes, no replication in the offspring. And a good example of one of these somatic mutations would be things like cancerous tumours, for example. If, however, the mutation is associated with meiosis, and obviously that's the process that's going to be producing gametes within the organisms, then that will potentially be inherited by the offspring. Because if in that gamete that's been created we've got a mutation, that then goes on to create some form of offspring, that offspring will then have the mutation present. So, mutation of the gametes will be inherited by offspring. Mutation of our normal body cells will not be inherited by our offspring. One of the things to bear in mind here, and one of the links that we can see happening in terms of the questions we get on the exam paper, is when we're talking about these gene mutations, we're talking about a change in that base sequence of the DNA. And what we are going to be able to link that back to in our work on Module 2 is the whole protein synthesis process. Because if we're changing the actual base sequence in that DNA, we're potentially changing those triplet codons. The triplet codons code for particular amino acids, and if we're then obviously changing the sequence of the amino acids, we will then change how that protein is going to eventually end up in its three-dimensional shape. So what we're seeing here is we could see just minor alterations, or they could be very significant. And if the protein that we are synthesizing happens to be an enzyme, for example, then we can link that into the ideas about alterations to that tertiary structure, leading to change of the active site shape, and therefore it no longer being complementary to the substrate. So whatever reaction it's catalyzing will no longer be catalyzed. If we now delve a bit deeper into the types of mutations, we can divide them up into two categories. Point mutations, where a single base pair is being substituted for another, and indel mutations, which are where we've got one or more nucleotides being either inserted or deleted from the DNA. and this leads to something we call a frame shift. Let's start off with the point mutations first of all, and these point mutations are one of three types: silent, missense, or nonsense. Let's start off with the silent point mutation then. Before we delve into what this is actually going to mean, we need to understand this word degenerate. And when we're talking about the genetic code, we can describe it as being degenerate. When we use that phrase as far as the genetic code goes, this just tells us that we've got more than one triplet code that is able to code for a particular amino acid. So when we have a look at our little diagrams that hopefully you're familiar with, so we've got the little wheel version, you can see tables, etc. Then what we can see here, if we have a look at our alanine here, for example, then we start in the middle, so we've got G, then C, and U. So that's one triplet that would code for alanine. However, we also have GCC, GCA, and GCG that all code for alanine. So what we could see there is that if one base is substituted, so if we're substituting that third base in our little triplet codon, what we can see is it's still going to code for that same amino acid. Therefore, that single base substitution hasn't actually changed the protein structure, because if we've got the same amino acid being coded for, our primary structure is going to be the same, which means the secondary structure is the same, and the tertiary structure is the same. No level of protein structure is altered with that silent mutation. The second point mutation that could occur is what we call a missense mutation. And what we're seeing here is that this little change in the base sequence is going to lead to a change in the amino acid sequence. So because we are changing that codon and therefore a different amino acid is coming in, this means the primary structure of that protein is altered. That may mean the secondary and tertiary structures change, but it's not a guarantee, remember, because obviously your secondary structure, which is your alpha helix or your beta pleated sheet, and your tertiary structure, the way it all folds together based on these interactions, if the amino acid that we've swapped is very similar in its structure to the one that we were meant to have, we may not actually see those changes to secondary or tertiary, but it's a possibility. Good example of this is sickle cell anemia. So this is a missense mutation on the beta polypeptide chain for the hemoglobin. And what we see is we're just changing the glutamic acid for valine. So a single amino acid switch has taken place as the result of a change in that codon. So what we then find is that one amino acid being swapped is going to mean that we end up with this sickle shape of our hemoglobin. And that obviously interferes with our ability to transport oxygen. So we can see that that one obviously has quite a significant effect. Third type of point mutation is the nonsense mutation. And this is where that point mutation has resulted in us creating a stop codon as opposed to anything else. Now, if we've got a stop codon, basically that means that wherever it occurs within our little sequence, it's going to stop the protein being synthesized there, because the stop codons just mean we've reached the end. So as a result of this, what we're likely to form is what we could term a truncated protein, i.e. a shorter protein than it should be. Some bits are missing. As a result of that, it may not be able to carry out its intended function. And so you've got Duchenne muscular dystrophy as an example of one of these nonsense mutations. So the second category of mutation are the indel mutations. Now indel, literally just split that word up, the in for insertion, del for deletion. So we are literally going to insert or delete something from this base sequence, and as a result we get what we call a frame shift. So when we think about our little frames, then what we end up with is these little sequences of 3 triplets. So that is an individual reading frame, that triplet codon. If obviously what we're going to do is insert or delete a single base, so if we were to insert another base before that A, what we're going to do is move that reading frame. We're going to change the codons. Same idea if we were to delete it. It moves that little reading frame to a different place. And the key thing about these Indel mutations is that if we are just inserting or deleting one or two little bases into our sequence, then every single base triplet that comes after that point is going to be altered. And that's because our DNA is non-overlapping. So that just tells us that each base is read only once, and these adjacent codons do not overlap with each other. Therefore, if we are inserting or deleting a base or a couple of bases here, then we are going to cause an alteration to all triplets that come after that point. So that's what we mean by a frame shift, the alteration of all of these subsequent triplet codons, meaning this is a big change. We're going to see a change to the primary structure because different amino acids will be coded for. We are going to then see significant impacts on secondary and tertiary structure because it's changing all of those amino acids. If, however, we insert or delete an entire triplet of bases, then what we're not going to see is a frame shift. Because if we've obviously got, and I'm just going to pick a random little code, oh, let's do that. There we go. So if we've got this little sequence of bases, then we've got our three little codons there. If we were to obviously delete the one in the middle, we don't get a frame shift because we've deleted an entire codon. So none of the others around it are going to change. We've just lost that amino acid from our sequence. Obviously, same thing can happen if we're inserting. If it's a triplet that we've inserted, it will not cause a frame shift. It will just have an extra amino acid present. Now, in terms of some of the genes within the genome, we will find that they've got these things we call repeating triplet codes. So a good example of this is Huntington's disease. And if you look at the Huntington's disease gene, then we find that it's got these things called CAG repeats. So just this repeating sequence of cytosine, adenine and guanine. Now, what we actually find is that this is an example of what we term an expanding triple nucleotide repeat. Because what happens is, during meiosis, we're going to actually increase the number of those CAG repeats. So this is one of those things that we see in terms of Huntington's disease, that it used to be thought of as the disease of middle-aged people, so you wouldn't expect to see any symptoms until you're in your 40s, etc. But we now understand that the greater the number of those CAG repeats, then the earlier the onset of the symptoms will be. And this is why these days in families that do have Huntington's disease, we are seeing juvenile Huntington's. So that's where it's got its onset when they are still incredibly young. So obviously not pleasant disease. Certainly hope that it's not in anyone's families, because it is something we cannot cure in any way, shape or form. We can merely treat some of the symptoms, but it's not going to be fixing anything, and it is progressive at the end of the day. The last thing that we're going to mention here is just a reminder. Even though all these mutations we've looked at so far have been harmful, not all mutations are harmful. We have neutral ones. These are the ones that don't really have any positive or negative impact on your life, just things that are. Things like the shape of your earlobe. You are not more likely to survive or die as a result of whatever shape your earlobe is. It's just a different shape. The inability to smell honeysuckle. Again, not more likely to die, not more likely to survive. You just may or may not be able to smell honeysuckle. Doesn't have any real impact on your life, full stop. We then have the beneficial mutations. Now, a beneficial mutation is going to be one that does give some kind of advantage, and therefore what we've seen is through that process of natural selection, it's caused this evolutionary process to take place. And if we think about two examples linked into high sunlight intensity and humans, we've got the melanin concentration in the skin and iris pigmentation. So if we think about the iris pigmentation first, this is basically the colour in your eyes. Now, what we actually find is that if you've got a region of the planet where the sunlight intensity is very high, then you don't really want light coloured irises. because that's not acting as any form of protection, and therefore you're more likely to suffer with cataracts as a result of that UV exposure. However, if you were in some of the more temperate regions of the world, like good old England, and that's where you were evolving in the past, then it would actually be more beneficial to have these lighter coloured irises, because you could see better in these lower light levels. So different regions of the world clearly evolved people with different coloured irises, because it was more beneficial to have darker irises in those high sunlight intensity countries, and in the countries like good old England, it was more beneficial to have a lighter coloured iris. And that's what's actually caused those differences in eye colour over time. We then have the melanin concentration in the skin. Now, melanin is that pigment that causes all the different skin tones throughout the world. And what we see is a similar pattern. People with a darker skin tone have greater protection from the sun's ultraviolet rays. So what you see is people who've got dark skin don't have the same rates of skin cancer as those with very pale skin tones. So that all just comes down to the melanin concentration. So what we have a look at is those early humans in Africa. It would have been advantageous to have darker skin, because it would have been that natural protection from the higher intensity ultraviolet rays from the sun. They wouldn't have experienced the same levels of skin cancers pre-sun creams. If, however, you had much paler skin, then it's beneficial in countries like good old England again. We don't particularly have to worry about high sunlight intensity most days of the year, which means if you've got lighter coloured skin, that's going to mean that you can actually absorb enough sunlight to enable the body to make vitamin D and avoid conditions like rickets. If, however, you've got a very dark skin tone and you're in a country like ours, then that may actually prove to be a bit of a problem for you. So even though having a darker skin tone is beneficial in these countries with very high sunlight intensity, in a country where we've got these lower light intensities, because your skin has this natural protection, it's not absorbing the same amount of sunlight. Therefore, you may not be able to make enough vitamin D, and you might need to take supplements. So it's one of those things that the different regions of the world, we've all evolved in slightly different ways, because different changes in our genomes were advantageous based on our habitat at the time. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and head on over to the website where you can find a range of other resources to help you in your A-level biology studies.